Hello and welcome to my PhenoRob Graduate Talk today. I will give you a brief overview of what I did during my PhD project with the title Imaging Spatial and Temporal Soil Water Content Variation of the Soil Plant Continuum using ground penetrating radar. As we all know, our modern society is facing some global challenges. And if we want to ensure food security for future generations, we need to make agriculture more sustainable. This can include developing climate resilient crops, managing our soils in a sustainable way, and using natural resources efficiently. One way to achieve that is investigating the soil plant continuum, where increasing yield potential by reducing the environmental footprint are key aspects. We can achieve that by mapping soil heterogeneity, improving field root phenotyping, and identifying soil-plant interaction. The investigation of this soil-plant continuum ranges across different resolution and scales. And at the high resolution and point scale end of the spectrum, we have direct soil sampling and soil sensors. And at the higher end and at regional scale and low resolution end of the spectrum, we have airborne methods like drones and satellite. If we want to measure at a moderate resolution and at plot to field scale, geophysical methods come into place. And if we use them in an agricultural background, this field of research is called agrogeophysics. We have different methods in agrogeophysics, and they mainly differ in their measurement frequency. And the lower end of the spectrum, where we have direct current methods like ERT. And in the low to mid frequency range, we have inductive methods like IP, SEIT, and EMI. On the lower end of the spectrum, we have ground penetrating radar, what I used in my PhD. Because of this higher frequency, we have a wave propagation nature of this measurement type. And this is also which gives us the highest measurement resolution compared to this other method. Because of the wave nature of GPR, we can directly derive the permittivity, which can be transformed into soil water content and porosity. And we can also derive the electrical conductivity, which can be transformed into clay content and salinity. I will give you a few aspects about ground penetrating radar, which are important for this presentation. So depending on the medium our GPR wave is traveling through, we get different wave types. So in air, we have our direct air wave. On the boundary between air and soil, we have the direct ground wave, which is a refracted wave. And within the soil, if we have contrasting soil layers with a different permittivity, we get a reflected ground wave and a refracted ground wave. GPR has different measuring configurations. Within my work, I use zero offset profiling within boreholes, where we have a transmitter and a receiving antenna in neighboring boreholes. And we move those antennas simultaneously across the boreholes and measure the direct ground wave in between. Since we don't have a direct air wave and we need this for calibration, we do additional wide angle reflection and refraction measurements, where we have the transmitter at a fixed location and then increase the offset of the receiver. Important to know about GPR, we don't just measure this straight wave between the transmitter and the receiving antenna. We measure a whole soil volume, and that is shaped like an elongated rotational ellipsoid. And the volume of this ellipsoid is depending on the antenna spacing, the frequency that we're measuring in, but also the medium permittivity. So if we have a higher soil permittivity, our measuring volume is smaller. I will give you a quick overview on how I analyzed my data. So we did the zero offset profiling method, and therefore we first need to do a time zero correction. And with that corrected wave, we get a first break, and that gives us the travel time of our GPR wave. Since we know the distance between the two boreholes, we can calculate a velocity, and this velocity can then be transformed into our dielectric permittivity values. And if we use petrophysical or empirical relationships like tops and crims, we can calculate the soil water content. There are more sophisticated analysis methods which consider the whole wavelet, but I have not been using them so far. I showed you what we measure, but to consider the soil plant continuum, we need validation methods. And this is where our measurement platform, the mini rhizotron facilities in the housing come into place. They allow us to additionally take root images. So we have two rhizotron facilities, which are closely located next to each other, but they're located in contrasting soil conditions. So both are river rural sediments, and we have in the upper facility a very coarse-grained soil, and in the lower facility, a fine-grained soil. They are both covered by a 30-centimeter plow layer, like a regular agricultural field. Each facility consists of 3 plots, and these different plots allow us to compare different agricultural treatments. And we have the facilities since 2012 and 14. So different agricultural treatments were considered over the years, which included surface water treatment, soil density, sowing date, sowing crop type, and cultivar mixtures. Each plot is again constructed the same. We have horizontal rhizotubes, which are 7 meters long. They're located in 6 different depths with a horizontal spacing of 75 centimeters, and we have a set of 3 rhizotubes per depth. Our crops are always sown perpendicular to our rhizotubes, and they are transparent. And with that allows us to take root images. So we take 20 root images to either side of the rhizotubes, and we do our zero offset profile measurements using a 200 megahertz system. We measure in a horizontal spacing of 5 centimeters. We have additionally soil sensors installed in the rhizotron facility, and with the root images and the GPR data, that gives us a quite comprehensive data set. And since we are measuring since a couple of years, Felix Bauer and I published a data paper to get the data out so people can use it, and especially to enhance collaboration across different research fields. So I showed you what we measure and how we measure, and now I wanted to give you a brief overview of what I did with all this data. So the first part of my presentation will be on how to link GPR data and root information and how we can quantify and correlate that. This still left some question, especially since we measure at different volumes. So we did a synthetic GPR simulation study using GPR max. And in the third part of my presentation, I used the soil water content that was derived from GPR to derive the soil hydraulic parameters. So linking GPR and root information, since, as you can see here, if we plot our permittivity along the rhizotubes and the different depths for one day, we cannot really distinguish which permittivity values are connected to roots and which are not. So we came up with a method to analyze this, which is called the trend-corrected permittivity deviation. And the first step is to remove dynamic influences. What I mean by that, can you see here? So the different colors represent the dates throughout the crop growing season. And this is along the rhizotube. So as you can see here, especially if you consider the different dates, we have quite varying permittivity values. So we again cannot really see which permittivity values are influenced by root. So what we did to remove the dynamic influences, we took the mean along the rhizotube and subtracted that from each zero offset profiling position. And this looks like this. And as you can see here now, we can see some deviations along the rhizotube, but there's a trend present. So we did this separately for the vegetation period and for the bare field value, so which before anything was sown on the facilities. And here this trend is present, but not these variations. So we thought, so this is, so there we can see our deviations and also the soil heterogeneity. So we took the mean of these values and again subtracted that from our spatial permittivity deviations. And this gives us our trend-corrected spatial permittivity values. I will show you this in more detail on the next slide. And now I want to direct your attention to this plot. Here I added the route information that we have overlapping with the GPR data that we can use. And as you can see, that is only 10 positions. So spatial correlation is kind of difficult. Looking at that for one plot, for one facility, for the different depths. And again, the different colors are the different dates throughout the crop growing season. And as you can see, we have these maxima and minima present along the rhizotubes. They have changing amplitudes throughout the crop growing season, so we can definitely say these are influenced by the crop. Here, for example, we have an increase throughout the crop growing season and then a slight decrease towards the end of the growing season. And if we compare that to our 1D root volume fraction values, so this is the mean of the root volume fraction along the rhizotubes, since we only measure on the edge of the rhizotubes. So you can see here that we have this increase throughout the crop growing season and then a decrease towards the end that we can see here. And we can see the clearest minima and maxima along the rhizotubes and the depths where we have the most roots present. To quantify and correlate the two values, we needed to come up with a way to find a 1D value for these trend-corrected permittivities. So in the next step, we calculated the probability density functions, which are plotted here, and their shape changes over the crop growing season. So we have a narrow peak early in the growing season, and this extends towards the end. And this peak can be quantified by the standard deviation, and that's will also give us again like a depth profile like I showed you before for the root volume fraction. And this is what we use in the next step for our correlation that you can see here. So we have mostly, or we always have a positive correlation. And to give you a bit more insight into this, I counted the R squares that are above 0.5 for the 2 years for the two facilities separately and for the different depths. So this percentage represents of how many correlation pairs are above 0.5. And as you can see for the 1680 centimeter depths where we have a lot of roots, we also have a good correlation between the GPR and the root information. And in the lower depths, not so much. So there's still often a lot of information that we cannot really validate with our root information. And that's why we did in the next step our synthetic simulation study with GPR max. And here we used to have more spatial root information. We used a root trench count that was done in 2017. So there for a grid of 3.75 centimeters, the root count density was estimated. And from that, we again counted our root volume fraction values and interpolated that for a 1 by 1 centimeter grid. This is the result for the two facilities. So here you can again see the quite contrasting soil conditions. So we have a lot more roots and more distributed roots for the lower rhizotron facility, which had the fine grade soil. So from our root volume fraction values, we needed to calculate the permittivity as an input for our software called GPR max. Traditionally, that is done for a three-phase soil system, only considering the soil matrix, water, and air. And we added a fourth root phase to this calculation, where we considered the roots to grow into the pore space. And here are our equations. Additionally, we needed some information for our simulation, but also for the calculation of the permittivity, like the porosity of the soil, the permittivity for the water, the roots, and the soil. And the soil water content, we considered 3 different scenarios for the two facilities. And for the simulation, we additionally needed the electrical conductivity of the soil and the roots. To see the influence of the roots on our GPR signal, we considered 4 different scenarios. So one where we only had soil, one where we added the roots, and additionally we added an above ground shoot to maybe see an influence of that for the maize crops, since they're quite thick. But little spoiler, that didn't do much on our data, so I will only show you the first 2 scenarios. And within our model, we have basically a replica of our rhizotone facilities. So we have neighboring tubes in different depths, and they have a horizontal spacing of 75 centimeters. Before I compare the different scenarios, I told you in the beginning that we have a refracted air wave on the boundary of air and soil, and this is what you can see here. So the black line shows scenario 1, and if we don't have any roots, all the traces should look like the 1 meter and 1 meter 20 depth, but as you can see, it doesn't. So this is why our direct ground wave is overlapping with the reflected wave. So this messes up our first arrival time. This is also why I never showed you the 10 centimeter steps. So we have the most influence there. And also it messes up our amplitude as well. This, and also what I will show you next, is why we need more sophisticated analysis tools which consider the whole wavelet and not just the first arrival time, what I have been doing so far. If we consider the two scenarios, we see a change in our first arrival time. So we could do some analysis, but if we look at this phase shift that is present, for example, here in our first cycle maximum amplitude, and also in an arrival of the reflected wave later on, we see differences. So we really need to consider the whole wavelet and not just the first arrival time. For the 60 centimeter steps, I will show you this over a few time steps. So this is above here, not considering any roots. And as you can see here, the wave travels in a circular way throughout the soil. And if we consider roots, we have these scattering effects. I showed you in the beginning or earlier that we also calculated the soil water content for different scenarios, considering roots and not considering roots. So we took our root volume fraction along the depth and calculated the 3 and 4 phased soil water content. And as you can see, if we are considering roots, the soil water content is much lower. So if we only consider 3 phases, that would overestimate how much water is present in the soil than actually is. With the soil water content, this gives us the opportunity to calculate our soil hydraulic parameters. So this is the third part of my PhD, where we did a sequential hydrogeophysical inversion to derive the soil hydraulic parameters. We did that for a winter wheat crop field. And there we have our GPR measurements, which give us the soil water content. And we have a modeling loop. And there we have a 1D hydros model, which consists of our hydraulic parameters. And we update them in this inversion loop where until our modeled soil water content matches our measured soil water content. And this model is basically, again, a replica of our rhizotone facilities. So we have different soil layers, our observation tubes, and as input, we use the precipitation, leaf area index, potential evapotranspiration, and the rooting depths. On the lower boundary condition, we have a time variable groundwater table, since we know the groundwater is influencing our measurements. We tested different model setups. So we used single and dual porosity, and we used an additional soil layer. So we have 2 and 3 layers. The results look like this. So the different colors here are the different model setups. These are the different depths. And as you can see, like a visual comparison is not really that possible. These red circles are our GPR measurements. So we use the root mean square error and the Geiger information criterion, which penalizes if we have a lot of optimized parameters. So if the model is more complicated, we optimize more parameters, which is the case for the 3 soil layer and especially for the dual porosity model. In the next step, we needed to choose one best model. And if we only look at the AIC, the best model would be the single porosity 2 layer. But we know there is some dual porosity present in our soil. So we chose the dual porosity model since there's a weighting factor in there. And if that is close to 1 or 1, we still have a single porosity. So with that, we can cover single and dual porosity. And the next step of the study was, since we have values of the from the rhizotubes along them, we can derive a spatial distribution of our soil hydraulic parameters. So we have 61 measurements that we can use. And what we did 61 1D inversion, which gives us a distribution of our soil hydraulic parameters. Here are the results. So we derive the soil water retention function. And here you can see the influence of the dual porosity. So in the flow layer, we have a dual porosity present. You can see this by this dent. And in the subsoil layer, we don't. And you can see here that in the flow layer, we have quite a distribution of our soil hydraulic parameters. And we wanted to derive some values that other modeling techniques could be used. So we derived the field capacity, the permanent wilting point, and the difference of the two is the plant available water. We also wanted to see if upscaling is worth it. So in the first part, we used the mean soil water content along the rhizotubes to do our 1D inversion, and then we used the individual soil water content values. So here we can see the comparison with our soil characteristics. So the green line is the 1D inversion results. And as you can see, this does not overlap with our mean upscaling results. So here's, for example, the green line for the permanent wilting point, and this is not in the middle overlapping with our mean upscaling results. So it can be worth it to go through that effort to derive spatial soil hydraulic parameters, even though you only need one value. So I will give you a quick recap what was achieved during my PhD project. So the first part, I show you how we can link GPR and root information. I showed you a new way to visualize the variability in the permittivity in the soil, which is influenced by crop roots. I quantified this and cross-correlated the GPR data and the root information. The second part, I showed you a basic GPR modeling study with the GPR response to root distribution. And from that, we took away that the zero offset profiling method that we're using at the moment is not sufficient, maybe in the future. And I showed you that it is necessary to include roots in the soil water content variation. The third part, I showed you the soil hydraulic parameter estimation for a crop field and showed you the benefits of that it could be necessary to derive the soil characteristic for a 2D distribution. Taking all that, where we can go from here, we could use surface GPR measurements or couple them with cross-source, since not everybody has a rhizotron facility on hand, of course. We need to measure with higher frequency to increase our resolution, since roots are kind of small scale. We need more sophisticated analysis tools like the full waveform inversion, like I showed you. And we need to enhance validation methods. So we still have this large offset between our root images, which are really small scale on the edge of the rhizotubes, and our GPR measurements, which measure a whole soil volume. We can couple our geophysical models with other modeling solutions to enhance field sampling, to maybe try test out field sampling methods synthetically before we go into the field, because that is a lot of effort. And also this coupling could enhance process understanding across different scales. And I wanted to take a few seconds to thank all the people who are involved in the different measurement campaigns and the support and technical maintenance of the mini rhizotron facilities. And with that, I'm already at the end of my presentation. I thank you for your attention